Good morning everyone. Welcome back to Ileum Biology. Today let's start with the process called RNA maturation. For that we need to clear some doubts regarding central dogma. What is central dogma? See, central dogma is a procedure. It's a phenomenon in which the information that is coded in DNA is converted into or it is expressed or translated into proteins. Let's see how it is done. First of all, the DNA present in nucleus gets transcribed into mRNA. The process is called as transcription. The message, the information that is present, that is coded inside DNA is first of all converted into mRNA. Now the mRNA which is formed is as per the DNA. It would be complementary to DNA. After formation of mRNA, the information which is present in mRNA is now converted into proteins, and the process is called as translation. And both these processes, transcription and translation, are together called as central dogma. In some organisms like retroviruses, for which HIV is a very good example, mRNA can also revert back into DNA, and this process is called as reverse transcription. That is reverse of transcription, and it is a phenomenon seen in retroviruses HIV. Let's start with the transcription first. I'll be dealing with the transcription in a very short way because our main topic today is mRNA maturation. But to understand the mRNA maturation, first we need to see what is transcription. See, in transcription, as I said earlier, DNA first gets transcribed into RNA. Now just after transcription, the mRNA which is formed is actually called as pre-mRNA, or you can call it as primary RNA transcript, or hnRNA, that is heterogeneous nuclear RNA. Why these terms are called so, we will discuss about it in the upcoming time. This pre-mRNA, primary RNA transcript, or hnRNA, is actually unstable, and by a process called as maturation, this unstable pre-mRNA gets matured into a functional mRNA, or it is called as exonic mRNA. This exonic mRNA is a stable molecule and is now ready for translation. So just after transcription, the mRNA which is formed is actually an unstable molecule called as pre-mRNA or primary RNA transcript, and hnRNA, which cannot translate into proteins. So it is first matured into functional mRNA or exonic mRNA, which is now ready for translation. So today in this video we are dealing with the process called as maturation. Let's start with it. First, let's see what is a split gene. See, if I talk about a split gene has got two components. One of them is a functional part and the other is a non-functional part. By functional part, I mean we are talking about exon, and by non-functional part, I mean intron. Exon is the functional part of gene. See, I am very clear here that both exons and introns would be transcribed. Functional part or non-functional part doesn't mean that one of them would transcribe and the other won't. Both of them would transcribe, but the exon, which when transcribes, it forms the part of mRNA which gets translated into protein, but the part of mRNA formed from intron won't translate into protein. So I can say that exon forms the coding part of mRNA and intron forms the non-coding part of mRNA. When mRNA is formed from a split gene, that mRNA would have two parts, a coding part which is formed from exon and a non-coding part which is formed from intron. The coding part gets translated and the non-coding part won't get translated. Now there is always a confusion between exon and intron, whether exon would code or the intron would code. It creates a confusion in the mind. So to clear that confusion, or to remember the difference, you need to remember only one thing, and that is exons express, EX for exons, EX for express. That means exons would express into proteins, but introns won't express into proteins. This is how I have remembered the difference between exon and intron. Let's discuss it with a very good diagram here. I'll be talking about the maturation of pre-mRNA and hnRNA now. See, in exon, let's see this diagram. This is a DNA with two ends, 3' end and 5' end. In this case, if you see, there are exons and introns. Exon 1, intron 1, exon 2, intron 2, and exon 3. This is DNA, and this split gene is now ready for transcription. When it gets transcribed, the exon 1 will form the coding first coding part, intron 1 will form the first non-coding part, and so on. But this is pre-mRNA, and if you see it clearly when you see our videos on transcription, and you will realize that against the 3' end of DNA, the 5' end of mRNA is formed, and against the 5' end, always 3' end of mRNA is formed. That means they both are complementary to each other. The RNA which is formed from DNA is complementary to DNA, so that's why the ends are complementary to each other. In split gene, the exon 1 was present near at the 3rd' end, but in RNA it is present on the 5' end. So that's how an hnRNA is formed. Now if I talk about the word hnRNA, heterogeneous nuclear RNA, why it is called as heterogeneous? See, hetero is a word which means different and homo means same. So why it is called as heterogeneous? Because this RNA has got two different parts, exon and intron, the coding part and the non-coding part. That's why it is called as heterogeneous nuclear RNA. Now this hnRNA is ready for maturation. So if I start with the maturation process, first of all, what happens at the 5' end of hnRNA, a cap of 7-methylguanosine is attached at the 5' end. 7-methylguanosine is chemically modified GTP, guanosyl triphosphate. It is a nucleotide, but this is an unusual nucleotide. See, if I talk about the usual nucleotides, they are A, T, G and C. But methylguanosine, guanosine is a usual nucleotide, but methylguanosine is an unusual nucleotide. So this is 7-mg, 7-methylguanosine, which is attached at the 5' end. Now see, let's assume that this hnRNA will now get translated into proteins. So if we start with the translation process from here, let's start with the process. It is starting from the exon 1, it proceeds in the forward direction, and when it reaches the intron part, this intron is non-coding part, so it won't code for any amino acid or any protein. So the process of translation gets terminated here, and the exon 2 or even exon 3 won't get translated. So this is not possible. That's why. So in this maturation process, introns are removed and the remaining exons are then rejoined. Intron 1 and intron 2 both are removed, and the remaining exon 1, exon 2 and exon 3 are then rejoined, and at the 3' end, as you can see, a tail of polyA. That means what is A? This is adenylic acid, the a type of nucleotide. This 200 to 300 molecules of adenylic acid are attached on 3' end, and this process is called as tailing. So mRNA maturation consists of 3 steps. In the very first step, which is called as capping, a cap-like structure of 7-methylguanosine is attached at the 5' end of hnRNA, and in the subsequent step, in the next step, introns are removed and later on all the exons are rejoined. This process is called as splicing. Splicing, it's a word which means to cut out. So introns are cut out and exons are rejoined, and in the next step, at the 3' end, a tail of polyA molecules, nucleotides, are attached, and this process is called as tailing. And now this mRNA is called as exonic mRNA. As the name suggests, exonic mRNA, it consists of only and only exons. No introns are present in exonic mRNA. So this is maturation of pre-mRNA, and this is a stable molecule. Now I will discuss these three steps: capping, tailing and splicing in a single slide. See, capping, it occurs at the 5' end first of all. It is a cap-like structure of 7-methylguanosine, as we studied. 7-methylguanosine is a chemically modified GTP, and the process capping is done with the help of an enzyme called as guanyl transferase. As I have already told you that the name of the enzymes depend on the substrate and the process it does. So this is called as guanyl transferase because it transfers guanyl, that is guanosine. Next step is the tailing step. It occurs at the 3' end of mRNA, and a polyA adenine tail of 200 to 300 nucleotides of adenine. The nucleotides of adenine are actually called as adenylic acid. So I can say 200 to 300 molecules of adenylic acid are attached at the 3' and the process is called as polyadenylation. Poly means multiple, polyadenylation because multiple molecules of adenylic acid are attaching themselves. So it is called as polyadenylation, and it is done with the help of an enzyme called as polyA polymerase. You need to remember the names of the enzyme. polyA polymerase is responsible for tailing and guanyl transferase is responsible for capping. Now many students get confused whether capping occurs at the 5' end or at the 3' end, and similar in tailing. So I am giving you a very good trick to remember this. You just need to remember T for tailing and T for 3' end. T for tail and T for third end. Third end means 3rd' end. So tailing occurs at the 3' end, and obviously capping would occur at the 5' end. Now if we talk about splicing, in splicing introns are removed and the exons are rejoined by RNA ligase. What is a ligase? A ligase is an enzyme which helps in joining. A DNA ligase joins DNA and a RNA ligase joins RNA. This process is accomplished with the help of some specific proteins which are called as hnRNPs. If you pronounce it, the word hnRNPs, it is pronounced as SNPs. hnRNPs, SNPs. And what is the full form of SNPs? It is small nuclear ribonucleoproteins. SNPs is small nuclear ribonucleoprotein. Let's see how splicing occurs by a very good simple diagram here. I am starting with pre-mRNA. In this pre-mRNA, there is an exon 1, intron and exon 2, and these are small nuclear ribonucleoproteins. hnRNPs, they attach themselves to introns. They will attach at, see, this intron has got two ends, a 5' end and a 3' end. So these hnRNPs or SNPs get attached to 5th' and the 3rd' end of the introns, and the introns get bent, and all SNPs together with some proteins, some other proteins, they form a structure which is called as spliceosome. So this structure in which multiple hnRNPs are attached together is called as spliceosome, and it is made up of SNPs and some other proteins. Now this spliceosome will cleave the intron from both of its ends, the 5' end and the 3' end, and subsequently the spliceosome will detach, taking away the intron, and the remaining exons are then rejoined. The spliceosome uses the energy from ATP to cut the introns. As you can see, these are exon 1 and exon 2. Intron is removed here, hnRNPs gets detached, and these exons are then rejoined by RNA ligase, and now this mRNA is called as exonic mRNA, and now it is ready for the process of translation. So that's how splicing occurs. Now I'll talk about some very good concepts that are many times asked in exams. See, gene has got difference in prokaryotes and eukaryotes. If I talk about prokaryotes, in prokaryotes genes are non-split genes. That means prokaryotes don't have any concept of exons and introns. They are not different. Prokaryotes always have non-split genes. But if I talk about eukaryotes, then eukaryotes have got split genes. That means a gene which has intron as well as exon. But there are exceptions. The two genes in prokaryotes which code for histone and interferon proteins are non-split genes. You need to remember this. Genes for histone and interferons are non-split genes. Rest all the genes in eukaryotes are split genes, but prokaryotic genes are always non-split genes. Now, RNA maturation occurs only in eukaryotes. This is very conceptual. See, in RNA maturation, there is cutting of introns. Are introns present in prokaryotes? No, they don't have introns. Prokaryotes don't have introns. If they don't have introns, what would be the significance of RNA maturation? So I am saying you need to remember this, that RNA maturation occurs only and only in eukaryotes. It does not occur in prokaryotes. See, split gene is an ancient feature of gene. It is not an advanced feature. It is a primitive feature. When you study the classification and evolution, you counter some words like in ancient characters or primitive characters, advanced characters. So split gene is a primitive character of gene, and presence of introns so is a primitive character and not an advanced character, but it is present in eukaryotes and not in prokaryotes. Can you relate what am I saying? See, prokaryotes are primitive than eukaryotes, but introns being a primitive character is present in eukaryotes rather than prokaryotes. So introns and RNA splicing both represents dominance of RNA world. What is RNA world? If we talk about the evolution, then RNA was the first genetic material which was later replaced by DNA because DNA was more stable. So the entire era in which RNA was the main genetic world, RNA was the main genetic material is called as RNA world, and the era where DNA is the main genetic material is called as DNA world. But RNA splicing and introns, they show the dominance of RNA. Now we will be discussing about the NCERT book. In the NCERT book of biology class 12th, chapter number 6th, where they have discussed about transcription. At the end of the transcription, we are taking this extract from NCERT so that I can explain the lines, the language of the NCERT. In eukaryotes, there are two additional complexities. Now, NCERT is comparing the process of transcription amongst prokaryotes and eukaryotes. In eukaryotes, there are two complexities which is not present in prokaryotes. The first complexity is that in prokaryotes, there is only a single RNA polymerase which is responsible for transcription of all the types of RNA, whether tRNA, mRNA or rRNA, whatever be the type of RNA, all of them are transcribed with the help of a single enzyme called as RNA polymerase. But in eukaryotes, it is not like this. In eukaryotes, there are clearly three different types of RNA polymerase first, second and third, and there is a clear-cut division of labor. That means each RNA polymerase has been assigned a specific work. There are at least three RNA polymerases in the nucleus, in addition to the RNA polymerase found in the organelles. In the organelles, by this line, they mean that even RNA polymerases are present in mitochondria and chloroplast, which are organelles. And other than those RNA polymerases, there are more three RNA polymerases in nucleus, and there is a clear-cut division of labor. Now we will be seeing which RNA polymerase does what type of job. RNA polymerase first is responsible for transcription of rRNA, that means ribosomal RNA, 28S, 18S and 5.8S. RNA polymerase third is responsible for transcription of transfer RNA, 5SR rRNA and snRNAs, that is small nuclear RNAs, and the RNA polymerase second is responsible for the transcription of precursor of mRNA, the heterogeneous nuclear RNA. Now you need to remember these clear-cut functions of different RNA polymerases. Now I'll be talking about the second complexity. See, we have discussed all these things, but why I have taken this extract from NCERT? Because many students say to me that, sir, this language of NCERT is a bit difficult. So I am just trying to simplify the language. The second complexity is that the primary transcripts contain both the exons and the introns and are non-functional. The introns are non-functional. As I have discussed, the primary transcript has got two parts, exons and introns are non-functional. So this is subjected to a process called as splicing, where the introns are removed and exons are joined in a defined particular order. As I have discussed, removal of the introns followed by rejoining of the exons. hnRNA undergoes an additional process called as capping and tailing. In capping, an unusual nucleotide, as I have already told you, unusual nucleotide methylguanosine triphosphate is added to the 5' end of hnRNA, and in tailing, adenylate residues around 200 to 300 are added at 3' end in a template independent manner. Now this fully processed hnRNA, which is now called as mRNA, is transported out of the nucleus for translation. That means it is ready. And where does translation occur? Translation occur in ribosomes, which is outside nucleus. So this mRNA is now transported out of nucleus by nuclear force. Let's discuss the figure given in NCERT. They have clearly given a very good figure which is self-explanatory. This is DNA with 2 strands, 5' strand, and the other is complementary to it, which is 3' end. In the very first step, this is RNA polymerase enzyme, and this is transcribing a mRNA. This is mRNA. This portion is mRNA. This is the 5' end of mRNA, and the moment it starts forming, at the 5' end, 7-methylguanosine cap is attached. This is the cap of 7 mg. And you can see these yellow ones are exons and these orange ones are introns. So we need to remove the introns. As you can see, the labelings exon, intron, this is at the 5' end, methylguanosine cap, this is 3' end. These are spliceosome, as you can see in the figure. These are spliceosome, and all of them are get attached to introns to cleave them, to cut them out. And at the 3' end, they have shown a polyA tail in green color, and the process is called as polyadenylation. And this is RNA splicing, wherein the spliceosome will cleave the introns, and at the end what is remaining is a cap at 5' end, exons, and a polyA tail, and this forms the messenger RNA. In the last paragraph, what they have mentioned is that the significance of such complexities is now beginning to be understood. The split gene arrangements represent notably an ancient feature of the genome. As I have already discussed, the split gene is a type of primitive character. The presence of introns is reminiscent of antiquity. What does antiquity means? Antiquity means ancient times. So this presence of introns is a remnant of ancient times, and the process of splicing represents the dominance of RNA world, as we have already discussed it. And in recent times, the understanding of RNA and RNA dependent processes in the living system have assumed more importance. See, if you want to clear the exams of NEET, what you need to do is to read the NCERT book thoroughly. Each and every line, each and every word is important here in NCERT. So clear them all, do not keep any doubt, ask your teachers so that you can get maximum concepts in your mind. Thank you very much for watching us. Do subscribe us so that you can get notified about our upcoming videos as soon as they are released. Thank you.